Okay, so let's just quickly do an overview of the stuff we've already done. So here we just got to remind ourselves of the basics. We want to make sure that we remember that we have these important functional groups. They're called functional groups because they're sticking on the outside of the molecule, and they determine whether that molecule is going to work or not. Will that molecule function? If you have the wrong functional group in a spot, you're going to have a different effect on that molecule. It'll do something different, or it just won't do its intended job at all. So you have to make sure that when you're building a molecule, it's built correctly. What you're going to learn later on is that the machines that end up building these things are made of protein, and it's the DNA that is the code for making the machines, and then the machines put these functional groups in the right spot on molecules when you're building molecules in the cell. So there's this master code in the DNA, creates the machines, and then the machines then go and create these molecules for us. Some of these molecules we're just getting in our diet because we can't make them ourselves, but we'll get to that when we talk about proteins in the next little bit. So make sure that you for sure are able to recognize a hydroxyl, a carboxyl, the amino, and the phosphate groups. Those are the most important ones. Sulfhydryl, I'll just sort of mention when we get to proteins, and carbonyl, I'm not going to focus on because it's not any of the functional groups where we're actually doing the joining of the molecules. Okay, then we ran through ionic bonding, and we said that an ionic bonding is when you transfer the electrons so that somebody becomes positive and somebody becomes negative, and then because they're oppositely charged, they will be attracted to one another. The ionic bond is a weaker version of intramolecular bonding. Intra because it's one molecule being put together. In fact, it doesn't even form a molecule. We wouldn't call it a molecule. We call it an ionic crystal. Because once I have the sodium and the chlorine ions, I can't guarantee that it's going to be this exact sodium ion matching up with that exact chlorine. It could be any chlorine ion matching up with that sodium. So this is more of a ratio kind of a deal, whereas when you're doing covalent bonding, you know exactly who the partners are. That's why back in grade 9 and 10, you learned about the different naming systems. When you were naming NaCl, you were just naming it as sodium chloride because it's a ratio 1-to-1. For every one sodium ion, there is a chlorine ion. So if I have 10 sodium ions, I have 10 chlorine ions, and they're just kind of partnered up. Whereas if you have a covalent compound, it will form a molecule with exact proportions. So if you had NO3, it's a nitrogen with 3 oxygens every single time. So we have to acknowledge the fact that there's lots of versions that we call nitrogen oxide, but how many oxygens did you need? So it's nitrogen trioxide for NO3, and so on and so forth. So very exact proportions, whereas these are just whatever. So not a great strong bond, but the neat thing about it is that the sodium, where it was a very reactive metal that would have, if it fell on your skin, it would burn you because it would react with the water in your skin. And the chlorine would be a gas that would kill you. When you create the ions version of them and you have that transfer of electrons, they're now stable. So both the sodium ion and the chlorine ion are no longer going to react and cause the same, they lose their distinct properties the way that they had before. And now they can dissolve in water, and they can easily be separated again, and so on and so forth. You can put them back into crystals and all sorts of stuff. The reason that those ions are able to dissolve in water is because they are charged. And we did talk about the idea that water is, because of the nature of the oxygen being so electronegative, such an electron affinity electron hog, that it is going to be slightly negative because these electrons that belong to the hydrogen, although technically they should be shared equally with hydrogen and oxygen, they just don't get shared equally because this monster here steals them most of the time. So that means that our oxygen gets slightly negative and our hydrogen gets slightly positive. And then because there's this polar charge to it, poles meaning opposites, because we have that, then we can say that anything that has that slight polarness to it, it's going to be able to dissolve in the water, okay? And the reason being that when, it's not so much that it fills in the gaps, but when that sugar molecule comes in, let's say, so we said the monosaccharides and the disaccharides are soluble in water, they are slightly charged, and what they do is they come in, and it's like they kind of push this water molecule down, and they form their own little connection with that oxygen. And then this water molecule will form its connection with the underside of the sugar. Does that make sense? Kind of like it pushes them in and out of the way, so it sits among the water particles. And then because of that, we would say it has dissolved. Okay, they form hydrogen bonds with water. Hydrogen bonds are going to get very important as we begin to build these other molecules that we're talking about, especially the nucleic acids, but also the looking at the proteins and whatnot as well. We talked about the terms hydrophilic and hydrophobic. That's also important. Remember that if you're non-polar, it means you're not going to interact with water, so you're hydrophobic. If you are polar, just like water, you will interact with water, therefore you will be hydrophilic. So looking at the carbohydrates, we can say that we've got carbohydrate, so we've got carbon with water, and it's in a ratio of 2:1. And when we saw lipids were also carbon, hydrogen, and oxygen, but no ratio of 2:1. I'll show you the picture again in a second. Right? I told you that I would want you to memorize the 3 monosaccharides, glucose, fructose, and galactose. Memorize their names, memorize their formula, and memorize the idea of what a structural isomer is. There's a test question that asks you to define structural isomer and give me a real example. I'll give you the answer again to that. And it's that a structural isomer has the same molecular formula, same parts, but a different structural formula. So those parts are in different locations. Okay? That makes it an isomer. Same parts, different locations. And then you can use any of these as your examples. You can say glucose and fructose. You can say glucose and galactose, or galactose and fructose, whichever one you remember. You can even use the disaccharides that we talked about. So di meaning 2. So when we stuck those 2 simple sugars together, you have the exact same formula, C12, H22, O11, and you see there is still a 2:1 ratio there for the disaccharides because all carbs will have this 2:1. So it makes it a hydrate. So then we've got glucose plus fructose together are going to give us sucrose. And then glucose plus glucose give us maltose. And maltose is the one that I want you to memorize the drawing of and be able to repeat the drawing of, so that we're limiting what you have to memorize, but you still get the concept of how to build a molecule. So you'll need to know how to draw glucose, and then you'll need to know how to draw maltose. But you might need to calculate what the actual molecular formula would be for a trisaccharide. And then you just say, well, there's 3 of these guys, but I have to remove a couple waters. Every time you join them together, you remove 1 water, right? So if I had, let's say I was joining together 6 sugars, how many waters would I remove? 5. Okay, simple. And then again, we said that the mono and disaccharides, when we're throwing them into water, because the oxygen has way more influence in a smaller molecule, they will actually be ever so slightly polar, so they will dissolve in water. Whereas the big long sugars, overall, there's way fewer oxygens than there are carbons and hydrogens. So those little few oxygens don't have as big of an effect on pulling and making that big long molecule polar. So we would say overall, the big long starches are just non-polar. That's why they won't dissolve in water. We also said that the name of this, of how to join them, when you got these 2 hydroxyl groups, the name of joining these guys together, the reaction that they undergo is called dehydration synthesis, because I have to remove a water to join them. And then the other name for that exact same reaction is called condensation, because water is formed. And then I also asked you to memorize the idea that when you are dealing with sugars, you're putting the 2 monosaccharides together, whether it be glucose and fructose or glucose and glucose, that you have to acknowledge which functional groups are being used. And in this case, we have 2 hydroxyls that are being joined together. And then we have this linkage here called a glycosidic linkage. That is an important piece of information. So glycosidic linkage. So now when you think of carbohydrates, I want you to think of it's 2 hydroxyl groups, and when they join, they make a glycosidic linkage. I want you to memorize that in the context of building sugars. I also want to acknowledge the fact that there is a carbon over here and over here. I don't want that information to get lost as well. It is between the carbons. Then we discussed polysaccharides really briefly, and we said that there are the stored energy version of them. So stored energy versus the structural version. And so you need to know that the starch is the plant stored energy version. The glycogen is the animal stored energy version. And then that cellulose and chitin are plants and animals' structural version. I don't care if you remember 10 to 12 subunits versus 8 to 10. It really doesn't matter to me if you remember exactly how it branches like that. That's not a huge deal, all right? So don't worry about that. but you probably should have some information tucked away about the idea that cellulose forms a fiber. That'll help to create bulk, to help keep things moving within the digestive system. That's important. Polysaccharides are not soluble in water. Then we briefly went through lipids. And so just a reminder again, lipids are also made of carbon, hydrogen, and oxygen, but no 2-to-1 ratio. And lipids are going to be used as a longer-term energy storage molecule. They're also useful for other structural things. And we talked a little bit about that as well. We said as well that they do not form polymers. You'd never get a chain of fats. Instead, what you get is one fat sitting on top of another, and that's what I tried to simulate with the paper in the previous lesson. So you can get them sitting on top of each other, but you still have to build that single fat, that triglyceride. You need to take this glycerol, and then at each of these locations, you have an option of what you're going to add on to these hydroxyls. You're going to take a carboxyl end of whatever fatty acid you're using, and you're going to get rid of an O and an H from one end, and then just an H from the other end. And then this oxygen that's left over will then be free to join straight to the carbon that's left over. So when you do that, take out the H and this H and OH, you have H2O. So it's still a dehydration synthesis reaction. And then you can show it joined. So this is before and this is after. You can show it joined directly. These ones are actually pretty easy to draw once you get used to them. The one I will be testing you on is using butyric acid, and I'll show you what that looks like in the lipids activity that you were supposed to be doing. Remember as well that whereas carbs is two hydroxyls joining together through dehydration synthesis to make a glycosidic linkage, in this case it's a hydroxyl on the glycerol but a carboxyl on the fatty acid, and then you're going to form what's called an ester linkage. So you want to connect those ideas as well for lipids. And of course, if you're adding 3 fatty acids, if you use every single one of these hydroxyls, then you will be forming not 1, but 3 waters will form. So in your formation question at the very end, you want to say plus 3 waters. Then we talked about saturated versus unsaturated fats. And we said we call them saturated because if you look at the carbon strand, this hydrocarbon strand out here on the fatty acid, every location that could possibly bond with a carbon is filled up with the extra hydrogens, right? But if you look at an unsaturated fat, then you can see that rather than having a hydrogen sticking off, instead you've got this double bond to the other carbon because it's also missing a hydrogen. So here we have the saturated acid. Hopefully everything's still working here. Saturated acid, all full up of all of these, and I should say saturated fatty acid because this isn't a full fat. It's just the fatty acid part of the fat, right? All full up of hydrogens. Then here we have the cis double bond naturally occurring. And you can see we've got the hydrogens are on the same side, and that's what kinks the molecule. And then we kink again because the hydrogens are on the same side. But in a trans, one of them up, one of them down, it means that it's a little straighter of a molecule. You can see it visually now better, right? Okay. And so because of that, trans fats, by the way, some trans fats do actually occur naturally, but we also were able to manipulate fats, hydrogenate them, take these polyunsaturated, bubble in hydrogen, and be able to make these hydrogenated oils. And that's what formed the trans fats for us, okay? When we were trying to increase the life of the oils that we were using to fry stuff, right? Because we didn't want them to go rancid so quickly. So they last longer, but they behave because of their straight, almost 180 degree. They act just like one of these fats. Just keep going here. So then we talked about the idea that there are other types of fats out there, specifically waxes, which give you a nice, because they are non-polar, they'll be waterproofing for whatever you want to wipe a wax on. They have a very low melting point, but it's good enough to be solid, a soft solid at room temperature. You already know that because you've probably touched a candle before. Okay, it doesn't take much to melt a candle. Phospholipids are the ones we really care about for this particular course because they make up the cell membrane, and we're going to focus in on cell membranes and how cell membranes work. So remember then that you've got that same glycerol with the 3 carbons, but in the OH groups that stick off of it, one of them has this phosphate group that's sitting there. And if you look at a phosphate, that phosphate is surrounded by very unhappy, unsatisfied oxygens that are still greedy for more electrons. And so because of that, you get a charged head So you get a charged or a polar head that loves water. It's hydrophilic, versus these tails, which are nonpolar because they're such long hydrocarbon tails. Nonpolar tails don't like water, and so you get this automatic double layer that forms, or what we call a lipid bilayer, because it's made of fat. And you get the heads that stick out towards the water on the outside of the cell, so it'll be in this direction. And then you get on the inside where there is water in the cytoplasm, you'd also have the head sticking towards that, and then the little tails hide in together so that they don't have to make any contact with water. And that's our cell membrane at a glance. There's a whole lot else going on with it. We'll talk about it in a few lessons from now. So you have a polar head, nonpolar tail. You need to be able to describe the polar head as being hydrophilic and the nonpolar tail as being hydrophobic in your test. And then finally, we mentioned that there's also steroid hormones, which have a completely different shape. We're not these big long chains of things, but instead we're in carbon rings. And then whatever you stick on the outside of that ring, whatever functional group is there, if you change the functional group, you change what that hormone can do. And hormones are basically just molecules that are created by one cell in your body, one type of cell in your body, gets dumped into the blood, flows to other cells, and tells them what to do. It's a chemical messenger, and we'll do a whole unit on that later. So that should bring you up to date with how far we've gotten in our notes so far. Now let's talk about nucleic acids. So as I've written in the top portion there, nucleic acids are going to be covered in more detail. We're really going to go into them. This is just a really brief overview of what the general structure is of these huge macromolecules. But the important details are going to be like how we use it, what it all means, is going to be focused in on for an entire unit in another part of our course. Okay. So what are nucleic acids? Well, this is your DNA and your RNA. This is your genetic code. This is the code that makes proteins, as far as we can tell. There are huge chunks of DNA that do not code for proteins, and geneticists are still trying to figure out exactly what that means and exactly what those sections do. Some of them would regulate other parts, so they would, if they were accessible and turned on, then perhaps they influence another gene to say whether it can be turned on or off without making a protein product itself. We're still learning, got a lot to learn about genetics. We really only even discovered that DNA was the actual genetic material in the 50s. 1953 is when Watson and Crick discovered the possible structure of DNA, and only within the few years before them were they actually realizing, oh, okay, DNA is the thing that you inherit from your parents. Before then, they were very sure that it was proteins that your parents gave you, and that because your proteins look just like your parents. Turns out it's not the machine itself, it's not the protein itself that your parents give you. Your parents give you a code so you can make your own proteins. And again, so it's a pretty new science. You think it's only been 70 years. And in the 1990s, when we actually had the Human Genome Project, so we really haven't even known what the code was or what it stood for in its entirety for more than 30 years. So it's a pretty new science. Very interesting field to be in. Okay, so nucleic acids then, as far as this unit goes, what I'd like you to do is get the idea of the similarities and differences between DNA and RNA. So I've tried to list them for you here. So DNA then, the full name of it is deoxyribonucleic acid. And the reason it's called deoxy, if you were to just chop off this deoxy part and just say ribonucleic acid, you'd have RNA. The reason it's deoxy is because on the sugar, I'll scroll down here for you. See in this is a sugar right here. It looks similar to fructose. It's not fructose. It's not a C6H12O6 version of sugar. It's slightly different, but it's got that same kind of pentagon structure to it. At this one location, the functional group is different. For RNA, it's the full OH group, so it's an actual hydroxyl, but DNA doesn't have the O. So we call it deoxy. It's missing the oxygen. And that's one of the fundamental differences between RNA and DNA. It's built right into the name, so hopefully you won't forget that one. So you have a different sugar. It's a deoxyribose versus a ribose sugar. They all have a sugar, they all have a phosphate, and then they all have some kind of a base. That's called a nitrogenous base because you can see there's nitrogen built into the ring. There are 4 types of bases for DNA and 4 bases for RNA. And the order of the letters of these bases is going to actually be the code itself. that'll tell you what protein you can make out of this DNA. So you can see that there are 3 in common. They both have adenine, they both have guanine, they both have cytosine. But when it comes to DNA, you have something called thymine, which is this one right here. And then in RNA, it's replaced with uracil. But if you looked at the structure here of the thymine versus the uracil, you can see it's only different in one spot. One functional group is different. So you've got this little CH3 group over here in the DNA version, in the thymine, whereas that is missing here. We just have a little H sticking off. So again, functional groups matter. What you stick where on a molecule makes a big difference on how that molecule can behave and how the body can use that molecule. Functional groups matter. Okay, so that's another difference, is the fact that DNA has thymine and RNA has uracil. But I said I wanted you to do similarities and differences. So if you wanted to choose a similarity, you would say, well, they have 3 bases in common. Because this is a 12 university level course, when you give me your answers, you need to be very specific. So you can't just say they have bases in common. Tell me what those bases are. Give me their names. That's how you'll get your full marks in a 12U course. Okay, so you can't just be like, well, there's some things that are the same. Yeah, well, what are they? Tell me specifically. Show me that you know what you're talking about in your answers. You've got this huge big ladder shape going up, and this is a pretty iconic picture of DNA. This is usually how they show it, right? What you have to know, though, is that this ladder, even though in this picture it shows like it's one complete bar going across, it's not actually. In fact, the one side of the ladder is not actually connected to the other side. If you look at the picture down on the right, you can see here we've got the phosphate and the sugar and the base. So that's one, what we call a nucleotide. Okay. And then we have the next nucleotide here. These would be considered the monomers of the nucleic acids. And you can see that there is a physical join between the bottom of this sugar and this phosphate of the next nucleotide. Then obviously the phosphate gets joined to the sugar, then the sugar gets joined to phosphate, so it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And because it alternates like that, phosphates and sugars all the way down, we call that the sugar-phosphate backbone. Pretty simple. And then off of every sugar, you're going to have a base. What that base is depends on what you want your code to be. What kind of protein do you want? We'll get to that. But you notice that it also has that exactly the same on the other side. But you notice that the other side goes in the other direction. See how this sugar is pointing up, but this sugar is pointing down? We call this anti-parallel. So it actually runs in opposite directions. So we have phosphate, sugar, but now we've got phosphate, sugar going the other direction. That'll become more important in our next unit. The reason I wanted to come to this picture, though, is I wanted to show you that whereas this forms one half of the ladder and this forms the other half of the ladder, they are not actually physically joined to one another. They do not share electrons. There's no chemical bond holding them together. Instead, what we have is an attraction between the bases. It's called hydrogen bonding. That same idea where water becomes polar, that's happening here as well. These nitrogenous bases that are attached, let's go back down and look at them again. So here's an example here for us. These nitrogenous bases that are attached, these nitrogens, just very similar to oxygen, because they're not as electronegative as oxygen, but they're second best. They're pretty close. They're also a bit of an electron hog. So they will happily steal electrons from these hydrogens that are here. And if that's the case, the nitrogen becomes ever so slightly negative and the hydrogens become ever so slightly positive. And because that happens on both sides, both of the bases on either side of the ladder are doing that, you have slightly negative areas across from slightly positive areas, and then you have this clicking in attraction that happens. That's the hydrogen bonding. Between A and T, A always pairs up with T, and C always pairs up with G. Between A and T, there are 2 of these little bonds. And then between C and G, there are 3 hydrogen bonds. Memorize these ideas right here. Who pairs with whom? This is DNA, obviously, because we have a T here. And because it's double-stranded, RNA is most RNA is single-stranded. So if you want to do another difference, you could say that the DNA is double-stranded. Double-stranded meaning I've got 2 opposite things that are hydrogen bonded together. Versus RNA would be a single strand. DNA is also a huge, massive molecule, and an RNA is kind of like a snapshot. So imagine DNA is like the library, lots of books, lots of pages, and then imagine that the RNA is like a photocopy of one of those pages that you're allowed to remove from the library. That's the big difference between the sizes of those suckers. So hopefully that's enough information to make you feel comfortable with similarities and differences. And again, in another unit, we'll go into this in a lot more depth and try to get you guys feeling confident about it. Okay? And that's the end of the nucleic acids conversation.